Today I'm going to talk a little bit about some different bioactive metals, because Tim specifically asked me not to talk about iron. Apparently you guys do a lot of iron talks. So I picked a few other interesting metals that we have a lot of data on. I'm sure this is all very familiar, but just as a brief introduction, most of you guys probably know that the metals are very essential for phytoplankton. So I got a nice picture of a dividing diatom over here. And of course they need CO2 and sunlight. That's not enough. They also need nutrients like nitrate and phosphate. But also, of course, iron is really important, but it's not the only metal. There's many other metals they need as well. And of course, phytoplankton are the base of the marine food web and thus the driver of the ecosystem and the biological pump. And all required elements need to be taken from seawater. They have no roots. Everything, and they're not connected to the soil, so everything they need to take directly from seawater. So if we look at this is a very stripped down version of the periodic table. So the green ones are the biological elements, such as carbon, nitrogen, phosphorus, but also all these metals over here. So iron, of course, is well known, but they also need manganese, cobalt, nickel, copper, and zinc. And some of them have an absolute requirement for cadmium, but most don't. Calcium and silicate, of course, are used for the elemental, for the skeleton parts in some phytoplankton, like diatoms and crocodiliphores. And then there's still a whole bunch of elements that we know they have a linking with the biological cycle. They have a correlation quite often with the nutrients. But as far as we know, they have no biological function, so it's probably they're not involved in biology per se, but they're just hitchhiking along with the biological cycle. And today I'm going to talk a little bit about nickel, zinc, and cadmium. But cadmium has been known for a very long time to correlate with the major nutrient phosphate. Cadmium was actually one of the very first trace elements that we had accurate data for in the late 70s, early 80s. And immediately it was obvious that it correlated with phosphate. It was a bit of a surprise, because as far as we knew back then, cadmium had actually no role in biological cycles. As I just said, we found some phytoplankton species, diatoms mainly, that have an absolute requirement for cadmium, but most species absolutely not. So the underlying cause was immediately subject of debate on why is it correlating. Could of course be purposeful uptake for cadmium, implying there is a biological role for cadmium. We've identified a cadmium-specific carbonic anhydrase enzyme, so this is the enzyme that's needed to convert CO2, bicarbonate to CO2. But most phytoplankton don't have a cadmium-specific enzyme. But cadmium can substitute in the carbonic anhydrase, and then the enzyme still works. It only works less efficient, so it's about 70 to 80% as efficient. So on one hand, you can consider that a positive thing. If they run out of zinc, they can still use cadmium. But of course, when there was plenty of zinc around, you can also call it mild toxicity, because the enzyme doesn't work as well as it used to. Another theory is that it's non-specific uptake. So it's a case of mistaken identity. The phytoplankton are actually after some other bivalent metals, like iron or nickel or zinc. And they can't help it, but they're also taking up some of the cadmium. Either way, for this talk it doesn't really matter, because whether it's purposeful or non-specific, there is going to be uptake in the surface ocean and then subsequent remineralisation or regeneration while the particles settling down and being degraded by bacteria. So I'm going to talk about the geotraces section. So it's a GEO2 section that started close to Greenland. It was actually 3 legs, so we had a stopover at Bermuda, and Kristen was still there, so we had a nice visit. And then we went on to Brazil, and then we did the southern hemisphere from Punta Arenas to the equator. We had a bit of bad weather over here, so we had some holes, and we went back a year later to fill the holes when we were a guest on another cruise. And all in all, we hit 60 stations. The nutrients were done shipboard, and the metals were analyzed after the fact via multi-element offline pre-concentration and ICP-MS determination at the UC Santa Cruz. And we did an optimum multi-parameter analysis for the water mass distribution. I'll talk a little bit more about that, but it's just a tool to figure out which water masses are there at a certain part of the ocean. And it's based on the hydrological parameters, such as salinity and temperature, and the nutrients. So we sampled this with our NIOZ sampling system, which is quite a nice system. So when you sample for trace metals, you can't use conventional sampling systems. So at NIOZ we built our own. It looks like it's made out of metal, that's right, but it's made out of titanium, which is a very nice metal to work with at sea because it doesn't rust whatsoever. It still looks like today it was brand new. And these samplers are 23 liters each, and these are made out of PVDF, that's a Teflon-like material. Just like similar to GoFlows, only they have a different closing mechanism. And they close hydraulically, so it means you can't take these bottles off the frame. That's why we put the whole frame into its own clean room van. And you probably see that the color of the bottles has changed, because even though these look very nice and clean because they're white, they work very well for geotraces, but when we started collaborating with some biologists, we quickly found out we had a big problem because they're very transparent to light. Not so bad for us geotraces people, because the first thing we do is filter and kill everything that's in there. But if you also want to collect samples for bioassays or just general biological parameters, we found that it takes a little bit of time to get this out. You collect, for example, phytoplankton 20 meters. You bring it up, it lands on deck, you have to secure it and then push it in. It takes maybe 10-15 minutes. And in that time, enough light came through so that the phytoplankton got complete light shock. So we changed the bottles to polypropylene ones. They look a little bit grey dirty, but for trace metals they're still very clean. So in the van itself, we dress up in clean room suits, and then we can filter directly from the bottles. In this case, it is a PhD student of mine, Stanley, who's filtering a sample for iron isotopes. But I was going to talk about cadmium. So these are two ODV plots. I'm sure you guys have all seen ODV plots before. But we have a section. So 60 north is not too far from Iceland, so pretty close to Greenland, towards the equator. And then this is not too far from Punta Arenas in the south of the West Atlantic. Full depth, and the color scale indicates the color. So on top we have cadmium, and we can see that the distribution visually looks very similar to phosphate, but obviously the concentrations are very different. The maximum concentration of cadmium is just shy of a nanomolar, so 800-900 picomoles per kilogram, whereas the phosphate is 2.5 micromoles per kilogram. But the distribution pattern and the fact that they're depleted and elevated in different water masses is very similar. And you can immediately pick out the different water masses. And you can also use this OMP analysis for this. So basically it's an inversion. So you add an inverse calculation where you give the model certain end member values. You say, for example, Antarctic bottom water has a salinity of so and a temperature of so. And then you ask them, you do an inversion. So basically you ask the model, well, if you have all these degrees of freedom for this given sample, how much Antarctic bottom water do I have to mix with how much North Atlantic deep water to get the best solution? And of course, there's a large number of degrees of freedom, but Antarctic bottom water you're not going to find above, say, 2,500 meters. And all these other water masses, the South Atlantic subpolar mode water, also known as subantarctic mode water, Antarctic intermediate water, or upper circumpolar deep water, you're not going to find very deep. And North Atlantic deep water, also going to find in certain stations at a certain depth range. So for all these we could find a solution, and it basically tells you how much of a certain water mass is there. And it's very convenient when you want to unravel mixing versus biogeochemistry. So going back to cadmium phosphate, there's at least the suspicion of that they're coupled. They have a very strong correlation. But the correlation is also distinctly nonlinear, so it's not just a single correlation. This is a compilation by Jay Cullen in 2006. In the meantime, we have a lot more data, but I still think this is a very nice figure. Because people had fitted these two correlations through it, and basically it's a lot steeper in deep water masses than in surface water masses with lower concentrations. But if you do it like this, you can already see, well, even these correlations, they don't fit really well. You could have also tried to fit one line through it. And fitting the one line through it, that was what people really wanted to do, because they wanted to use cadmium as a proxy for phosphate in the past oceans. So if foraminifera, so they form shells, and they incorporate a little bit of cadmium in there, and basically you calculate the cadmium concentration in seawater used to be when the shell was formed, and people really want to calculate the amount of phosphate. And it's really nice if it's just one correlation, then I'm going to show you that's definitely not the case. So cadmium phosphate was used as a paleo proxy for nutrient conditions in past oceans, but of course if you have a non-linear relationship, like kink is changing the steepness of the slope, it's going to complicate the use as a proxy. So you want to know, why do we have the kink, and can we maybe come up with a way around it? If you know what side of the kink we are and we still have one correlation either side, we can still use it. So people were debating about the origin of the kink for quite a while. So it could be preferential remineralisation of phosphate, where phosphate is just because it's a required element. The bacteria really like phosphate, they don't really care about cadmium, so the phosphate is just remineralized quicker. But it could also be the influence of different water masses, or increased cadmium uptake under micronutrient should say the limitation, not remineralisation. So the idea is here, if, for example, phytoplankton gets iron stress, they want iron, they're going to upregulate all these transponders to take iron into the cell, and if there's no iron, but there's plenty of cadmium, hey, the mistaken identity, you get a lot more cadmium uptake. So this is the section now with, again, the blue dots are all the stations, so the 60 stations here. And we have all these different, so this is a very brief overview of the hydrography. So we have the subarctic gyre in the far north, we have both the subtropical gyres, the equatorial region, and of course the Antarctic circumpolar current coming through Great Passage here that we didn't sample. We were supposed to start here, but the ship we used had problems with their rudder, so we had to significantly shorten the section. So this is just a subset of the data. So here's a black bar. So this data is everything north of this black bar, so north of about 40 degrees, where we're in the subarctic gyre. And at first glance, it looks quite promising. We see quite a nice correlation between cadmium and phosphate. However, if we zoom in and use our EOMP model, we can actually see it's not best described by one correlation. It's actually 3 separate correlations. So we have cadmium versus phosphate. So now the color scale is North Atlantic Central Water. So we're just asking the EOMP model how much North Atlantic Central Water is in every sample. Of course, the deep samples, there's nothing. But in the surface, there's quite a bit of North Atlantic Central Water. It is basically the Northern Hemisphere subtropical gyre surface water. North Atlantic deep water, mainly in the deep, and subarctic gyre surface waters here in the middle. And then you can clearly see that in every individual water mass, or at least where you have the highest proportion of a certain water mass, that you have individual correlations. If we combine all data, it actually looks even worse. So this is all data for the entire transect. So, yes, if you really want to, you can fit like the not so steep slope and the steepest slope, but it's pretty obvious there's a lot going on. And I plotted the apparent oxygen utilization in color scale. So this is the amount of oxygen used for remineralisation. So it's a high number, that means there's a lot of remineralisation. And you can see that remineralisation causes some deviation both in the deep water, so the older Antarctic water masses, but also in the surface. You can't really see it, but I'm going to tell you this is the northern hemisphere and this lower is the southern hemisphere. So there's also a difference between the different hemispheres. And the funny thing is that this upper circumpolar deep water and Antarctic intermediate water, they look like they are different from Antarctic bottom water. But it's only in the northern hemisphere. If you only look at the southern hemisphere data, Antarctic bottom water, upper circumpolar deep water, and Antarctic intermediate water, they all plot along the same line. It's only when you get enhanced AOU, so when it's traveling northward, and there's particles raining into these water masses, because these are not too deep, the Antarctic bottom water is, but these two are not too deep, so there's particles raining into them. And a remineralisation causes this deviation from the single line. The famous king, you can see it, it's there, around phosphate of about 1.3 micromoles per kilogram. But there's a lot of other stuff going on, and like I just said, the subsurface water in the southern hemisphere are distinctly different from the northern hemisphere subsurface waters. So that begs, of course, the question, why? So this is a plot from the paper, and basically we could find not a single universal correlation, but we found many different, 8 correlations to be exact, that we could, with 8 correlations we could best describe the data set. And it was mainly mixing between the various water masses that leads to these many different relations. So it's just one water mass mixing with another, creating a mixing line, then another mixing line, and another mixing line. Don't expect you to read it, but these are the different, so sometimes it's just an individual water mass, but most often it's mixing between various water masses. So if mixing between the different water masses explained the kink and some of the scatter, but then we still had the fact that the water masses from Antarctic origin, they changed due to the interaction with the oxygen minimum zone, or when the AAU became high. So when there was a lot of remineralisation, that was another effect on top of the mixing. So this is where we can use the OMP for a more quantitative approach. So what you do with the OMP, you basically calculate the fractions of different water masses present for each sample. And then you can assign a cadmium concentration to each of the pure water mass. So cadmium is not actually part of this, and you solve this independently of cadmium just based on the hydrography and the major nutrients. And you say, okay, I'm just going to look Antarctic bottom water, I know where it is, and this is approximately the cadmium concentration in the pure water mass. You can just easily calculate what the cadmium concentration there should be by just multiplying the fraction with the concentration in the end member. And that then should give you what happens if there was only mixing. Well, we did just that, so we just estimated the end members and then run the model with mixing only. And this is measured phosphate versus the modeled cadmium. And this was the Antarctic bottom water, this is the North Atlantic deep water mixing. And these circles are still for the original data. So these were assigned not in the model, but in the by ourselves. And this is the line that was fitted to the actual, not the model, but the actual data. And you can see that the lines still fit the model data quite well. Proving that indeed, this kink is just the mixing between Antarctic bottom water and North Atlantic deep water. But you can also see that the model does quite a shitty job over here. So mixing only, not brilliant. So we get an R square of 0.95. It's not, a slope of 0.95 and an R square of 0.86, and a root mean square of 80 picomoles per kilogram. So it's not horrible, but it's definitely not as good as we wanted. However, we first optimized the mixing. So we asked the model, we estimated this. We said Antarctic bottom water is 800, but if we give you 10% degrees of freedom around that, what do you think it is? So 9 out of 10 times we were really close, but it mattered a little bit. But the biggest difference we got is when we asked the model to also infer remineralisation. And we already know how much based on the deficit of oxygen, and here we slightly redefined it. We call it the oxygen consumption, because the AOU assumes the oxygen concentration was saturated at 100% when the water mass was formed, and we didn't really care what happened outside the study region, so we basically reset the bar to 0 at the edges of our transect, so we only wanted to look at remineralisation along our transect. So we know how much remineralisation that was going on, so if we do a certain oxygen and cadmium remineralisation ratio, can you get a better fit? Well, definitely. We got an R square of 0.98, the root mean square error went down as well. And we came back with a remineralisation ratio of cadmium to phosphorus about 0.2 nanomole per micromole. So every micromole of phosphate remineralized would get about 0.21 nanomole of cadmium. And you see that improves the fit dramatically. So overall, the cadmium phosphate relationship is driven by mixing. That's the main. You already could explain 85% of the variability just by mixing. And the kink is caused by mixing of northern and southern origin water masses. And remineralisation within the Atlantic plains explains most of the remaining variability, of course not all, but most of it. So then we were wondering, is there then a constant cadmium to phosphate uptake and remineralisation ratio? If you want to use it as a proxy, well, this has to be a constant, otherwise it's really not usable. And we did see the different end members had very different compositions, so what explains that? And it's important to note the dissolved, so just dividing cadmium over phosphate, the dissolved concentrations changes as a function of the concentration as the intercept is not 0. So because of Redfield, we're used to the fact that for nitrate and phosphate, we can just divide one by the other to get the ratio. But it's not the case for cadmium phosphate, because I just took this black line just as an illustration. You can see when you divide cadmium over phosphate over here, you get 0.6 divided by 2, because this is picomole, it's 0.3. If you go down the same line, well, the ratio changes dramatically just because this has a non-zero intercept. So you can't just use the dissolved ratios, you have to be a little bit more clever about it. So mixing of end members is the most important factor, we established that, but still it doesn't explain the question, what explains the different end member compositions? Why is this so different from this? And does it affect how nutrients are supplied to the North Atlantic in general? So this is the cadmium to phosphate ratio. So I just said that you can't use that as a proxy for the slope, for the remineralisation ratio, but it is informative to see where do you have high cadmium with respect to phosphate. And we didn't calculate it for the very surface layer, because cadmium and phosphate have both almost depleted. So if you divide the number close to 0 by another number close to 0, it just goes to infinity. We didn't want to do that, so we excluded the top surface layer. And then if you realize that we're in the open ocean, so there is a little bit of supply from cadmium and phosphate, for example, via dust or from the continental margins. We were pretty much in the open ocean. And the supply of cadmium and phosphate is mainly coming from the deep ocean, the deep winter mixing and the diffusivity. And both cadmium and phosphate are depleted to pretty much 0 in the surface layer. Well, if that's the case, then if all supply is taken up, then the supply ratio must match the uptake or the average uptake and remineralisation ratio. If whatever you supply is taken up, well, the supply has to equal the uptake. Well, we can easily see what the uptake the supply ratio is just by looking just below the thermocline. So we defined the bottom as the thermocline is this second blue line over here, and you can see that in the southern hemisphere both the cadmium phosphate ratio as well as the actual cadmium concentrations are much higher. So that would imply that the cadmium phosphate supply and thus also the uptake ratio is a lot higher in the southern hemisphere than in the northern hemisphere, just purely because the waters that are supplying the nutrients to the surface ocean are higher here than here. We also found some evidence for higher cadmium phosphate uptake ratio in our model, but it wasn't significantly improved in the model, so we couldn't really take it. But it's an indication again that yes, indeed the remin ratio in the southern hemisphere is higher in the southern hemisphere compared to the northern hemisphere. But this leads to a different supply ratio, and that implies that it's not actually a universal ratio, it's just actually the supply of cadmium that determines the remineralisation ratio of the organic matter. And if you take it a little bit further, it's not that surprising. The North Atlantic deep water, so that's basically the one coming from the north, is mostly sourced from cadmium poor surface water, first transported north with the Gulf Stream to the high latitudes before becoming subject to deep water formation. That has a low cadmium and a low cadmium to phosphate ratio. Whereas the Antarctic waters that are mostly sourced from upward cadmium-rich deep waters, upper circumpolar deep water, has a very high cadmium concentration and also a relatively high cadmium to phosphate ratio. So then it's the ocean circulation that actually leads to variation in cadmium supply and uptake, and subsequently to variation in the end members formed in the different high latitude regions. So it's maybe a bit of a sobering realization that also implies that you can't actually use it as a proxy, because it's just going to change whenever the ocean circulation is changing, and of course with different position of the continents we know ocean circulation was very different in the past. So moving on to zinc. Zinc is also potentially used as a paleo proxy, because zinc actually correlates better with silicate. But there was a problem with this because we didn't actually know why silicate would correlate better with why zinc would correlate better with silicate than with phosphate. And it's not really makes no sense, because most of the zinc inside the cells, also in diet themselves, is actually associated with the enzymes and the intracellular machinery. It's only 1 to 3% of the zinc is actually in the silicon fructose. So we found out it's actually a combination of southern ocean processes and reversible scavenging of zinc of the settling and remineralizing material that made silicate correlate better with silicate, zinc better with silicate than with phosphate. So we did the exact same exercise as for cadmium, so spending a little bit less time on it, but you can clearly see here this is the zinc distribution that correlates better with silicate than with phosphate. No surprises there. You can see it just by eyeballing it. And when you plot zinc versus silicate, again for this section, the story is pretty similar. You can see that there is a kink, and there is just basically it's mixing between southern ocean water masses with northern hemisphere water masses, and of course the range of silicate and zinc is very large in the southern hemisphere water masses. You get very high concentrations in the Antarctic bottom water compared to, for example, North Atlantic deep water. So it looks like this is most of the data, but it's actually the split is about 50% here, 50% here. And again, strong influence of remineralization, because this again is a deficit of oxygen. So as soon as remineralization starts to occur, it deviates the relationships that we find. So we look here at the entire transect with now zinc versus phosphate. Well, it doesn't look like a brilliant correlation, but if you break it down to the various water masses, you can actually fit some quite decent correlations, but only when you select data. For example, you only look at Antarctic bottom water mixing with North Atlantic deep water, or North Atlantic deep water with North Atlantic subpolar mode water, you get pretty decent correlations. So again, mixing plays a very important role. If you only look at the northern hemisphere, it actually looks a lot better. So especially for zinc, even more so than for cadmium, there's a huge difference between the northern hemisphere and the southern hemisphere. So this is the northern hemisphere where you exclude everything south of the equator, and here's the other way around. Oh, sorry, it should have been the southern hemisphere, and everything north of the equator was excluded. So very different behavior of zinc with respect to phosphate. So we ran the same EMP model, and we tried to predict zinc by mixing only, and we expected the same. Like, we're going to get most of the going to be most of the way there, but we still get a significant improvement when we use remineralisation. Wasn't the case. We almost had 0.97 as an R square here and 0.98 here, so it barely improved the fit. The influence of remineralisation was not nearly as big as for cadmium. And it's not really surprising, based on the deficit of oxygen, it's about up to 200 micromoles in the oxygen minimum zones. The regeneration of a phosphate is about 1.2 micromole per kilogram. For cadmium, it's 0.25. The whole range of cadmium was only going up to 0.8. Whereas for zinc, you're getting about 0.5 nanomole back, but the range of zinc goes all the way up to almost 8. So yeah, getting another 0.5 thrown in here somewhere. Yes, it does improve, but it's down here. So for your fit, it doesn't really matter if these points are deviating a little bit. You're only adjusting them by 0.5 nanomolar. So in the grand scheme of things, this remineralisation is not as big as the influence of mixing. And the other interesting thing was that for cadmium, we saw that the supply in the southern hemisphere, we actually had quite a better supply of cadmium, more cadmium with respect to phosphate in the southern hemisphere than in the northern hemisphere. For zinc, it's the other way around. We have a very low supply actually of cadmium, just below the thermocline. So there's a lower ratio in the southern hemisphere than in the northern hemisphere. So this is raising again some questions. So it's interesting, they're different, and we can see where they're different. Doesn't quite explain why they're different. And most likely this zinc to phosphate supply and Riemann ratio is higher in the northern hemisphere, whereas for cadmium it was the opposite, the other way around. Then we started to look at nickel. So we parked these ideas, we figured out, well, they're different, but hadn't quite completely figured out why were they slightly different. But well, nickel, let's have a look at nickel. And it looks very similar to phosphate, very similar to cadmium. Nickel correlates with phosphate very well, correlates with cadmium very well, and it doesn't correlate with silicate as well. So basically the same story again, but maybe looking at nickel, a metal that behaves differently, will provide us with some answers. It actually did. Before we go there, this is the nickel in the upper 1500 meters. And nickel is different from the other bioessential metals. Both zinc and of course and cadmium, but also phosphate and nitrate, they go down all the way to pretty close to the detection limit in the surface ocean. The interesting thing about nickel is that the lowest concentrations we found were about 1.7 nanomolar. So it never goes all the way to zero. And the other interesting fact is that we find lower concentrations of nickel in the northern hemisphere subtropical gyre than in the southern hemisphere. Even though we know that especially in this region there's a lot of Saharan dust deposition, and elements like aluminium, also manganese and iron, they are slightly elevated over here just because there's more supply. Whereas nickel is lower over here. So that led to the hypothesis that there might be a role for diazotrophs. Diazotrophs, the nitrogen fixers, they have a higher requirement for nickel because it's needed in some of their enzymes that are related to the nitrogen fixation. And we know there is more nitrogen fixation in the northern hemisphere because there is more dust deposition, there is more iron, and iron stimulates the diazotrophs because they have a very high iron requirement as well. And so there's getting more iron, they're also getting a little bit more nickel through the dust, but because the diazotrophs are stimulated, they actually deplete the nickel to lower concentrations. Just a hypothesis that we hope to test in future research. Well, if we look again at the Riemann signal, so I already told it's about 1.2 for phosphate, 0.25 for cadmium, 0.5 for zinc, and surprisingly 0.9 for nickel. And why is that surprising? Well, if you look at the extended drug field ratios, as we call them, general phytoplankton, they should need more zinc than they should need nickel. However, we find that remineralisation in the Atlantic is stronger on an absolute basis for nickel than for zinc. So that could imply indeed that there is a lot of diazotrophs in the Atlantic that maybe have a higher than average requirement for nickel. And if you look at the grand scheme of things, so about 50% of the phosphate in the North Atlantic Ocean can be, in the regions with the highest concentrations can be due to Riemann. 30% of nickel, 30% of cadmium, 13 only of zinc, and only 6% of nickel. Because the nickel concentrations are quite high as well. So now we move to the Southern Ocean, because we still haven't answered the question why is there a lower supply of zinc in the Southern Hemisphere than of cadmium, and why is nickel again doing something differently? What we do know of the Atlantic Ocean is that the water masses that were formed at the high latitudes, North Atlantic deep water from the Nordic high latitudes, and Antarctic intermediate water, subantarctic mode water, and Arctic bottom water from the Southern Ocean, they play a really important role. And especially all this red blob of both for nickel and for phosphate and for cadmium, that's Antarctic intermediate water and subantarctic mode water. And they're formed, of course, in the Southern Ocean just south of our section. And this is a famous paper by Sarmiento et al., basically describing how nutrients from the Southern Ocean are fueling productivity at the lower latitudes. So we have circumpolar deep water coming up in the far south. Part of it goes to the continent and substick over there, and part of it starts to flow northwards into the frontal regions. So the circumpolar deep water starts flowing north, and then the Antarctic polar front, part of it subducts into Antarctic intermediate water. A fraction, of course, keeps flowing northward, and the other part subducts as subantarctic mode water. And because silicate this is a region where diatoms are dominant, so silicate is stripped out before phosphate and nitrate. So this water mass has almost, here there's no preformed silicate, yet there is preformed nitrogen, because there's nitrogen left over but no silicate. And this is often expressed as a negative silicate star. And also that's a reason why there's diatoms are not very dominant in the tropical regions, because there's almost no supply, because there's no silicate in this. And it comes to the same hypothesis that you have most of the supply is from deep waters or subsurface waters. There is nitrogen and phosphate supplied, but almost no silicate, so diatoms can't really thrive in those regions. So we wanted to see if maybe similar processes were responsible for the distribution of metals, and we think it is. So there's some data from the Southern Ocean, so this is just for your reference. Unfortunately, these figures have the latitude in the opposite direction, so south is here and north is there. Here it's south is there and north is there. But what I want to draw your attention to is first the nutrients. So the lower figure, you can clearly see that silicate, the green, is being drawn down much faster than phosphate or nitrate. So that's the reason why there's almost no silicate supply, because the water masses, the subsurface water masses that supply the nutrients are depleted in silicate, whereas phosphate is depleted much slower. If you compare cadmium and zinc, unfortunately there's no nickel data, but if you look at cadmium and zinc, we can actually see a very similar feature, that cadmium is only depleted from the polar frontal zone onwards, whereas zinc already starts to deplete here, and by the time you're in the subantarctic zone, zinc is already almost completely gone, whereas still half the cadmium is there. So zinc and silicate are depleted further south than cadmium and phosphate, and cadmium in turn is depleted faster than phosphate. And that's also the reason why these relationships has a non-zero intercept. So there's some very strong contrast between the zinc, cadmium and nickel supply in the North Atlantic Ocean, related to the southern origin water masses, where zinc is depleted in the far south due to uptake and export into deeper water masses with diatoms. And that leads to Antarctic intermediate water, and it's just a different name for the subantarctic mode water. They're both quite low in zinc, but 1.6, absolutely nothing, compared to the maximum concentration of zinc. So zinc is really depleted. If you look at cadmium and phosphate, depletion starts further north, so part of the export remineralizes in Antarctic intermediate water, and I'll illustrate it in the next figure. But that leads to this subantarctic mode water being relatively low, but Antarctic intermediate water being quite high, 0.1 and more than half of the total range. Nickel is the odd one out because it's never fully depleted, but we presume that uptake starts in the far south together with zinc, because diatoms have been shown by work from Twining that diatoms have quite a high requirement for nickel, higher than general phytoplankton. And there is a little bit of data from Andy Bowie that indeed nickel can be depleted quite low around the regions where we have high abundance of diatoms. But we really should go to traversing all these regions in the Southern Ocean and trying to measure nickel. But this is the similar figure, but now just for the metals. So we see that in the far south, zinc and silicate and nickel are depleted. The take-up starts in the far south, whereas cadmium and phosphate in the far south not so much. Then we get past the polar front, and Antarctic intermediate water is being formed. So that's relatively low, has a relatively low cadmium to phosphate, nickel to phosphate, and a zinc to phosphate ratio. Then zinc and silicate are stripped down completely. Nickel not all the way down, but it's stripped, and then cadmium only starts here, and phosphate as well. Then we go to the subantarctic zone. So zinc is completely depleted, silicate is almost completely depleted, nickel, well, they still take up some, but not a lot, and then cadmium and phosphate are depleted completely here. So that leads to this subantarctic mode water that has a low cadmium phosphate, a low zinc phosphate, and relatively elevated nickel phosphate, because phosphate gets completely down and nickel doesn't. But of course you have particles forming here, but they remineralize in this Antarctic immediate water that's below. So a lot of the cadmium, oh, the light just went out. So a lot of the cadmium and phosphate, they get remineralized in the Antarctic intermediate water. So even though there was a lot of depletion here, most of it is depleted here, so it returns back to the dissolved phase. Whereas for zinc and silicate, that's not the case, because by the time the water is here, there's nothing left. So remineralization will not return, right? That would have returned it in here. So zinc and silicate are caught in the Southern Ocean loop, whereas cadmium and phosphate, at least part of it, escapes. And nickel does something in between. It's partly in this loop, but because it never gets depleted completely, it looks like it behaves like cadmium, but it actually behaves more like zinc. Okay, and then this mixture of Antarctic intermediate water and that mode water that supplies nickel, cadmium and phosphate, but almost no zinc and silicate. That's generating these very stark contrasts between these correlations that we saw. The non-subsurface water masses also have a relatively high, low zinc to phosphate ratio, but not as low as the subsurface parts. And it implies that this process of depleting zinc before phosphate is not something that only happens in the Southern Ocean. It also happens in the Northern Hemisphere counterparts. Similar for cadmium phosphate, also the northern subsurface water masses have a relatively low cadmium to phosphate ratio. Again, depletion of cadmium before phosphate is not just something that happens in the Southern Ocean, it also happens in the Northern Hemisphere. But the big difference is we don't have both Antarctic intermediate water and subantarctic mode water. In the north we only have one of these water masses, subarctic mode water. There's no Antarctic intermediate water equivalent. So there is no loop trapping the nutrients, and that's the reason why cadmium behaves so differently. And like I said, nickel behaves more like zinc. It follows, it's also trapped in the Southern Ocean, but because the depletion is only partial, it ends up looking more, in the color plots, it ends up looking more like cadmium. So that's why I call them confounding correlations, because it actually took me several years to finally wrap my head around this. And definitely a take-home message is that the dissolved spot ratio, so just taking one over the other, is not representative of uptake and the remineralization ratio if the intercept of the relationship deviates from zero. So usually you can do it for nitrate and phosphate, but definitely not for most of the metals. And also a slope is not representative if the end members is not representative of uptake or remin ratio if the end members have a different composition and then mixing occurs. And because the observed correlation that all these slopes were very, very different, had nothing to do with the uptake and remin ratio. It was just the mixing between the different end members. Regional biological and chemical processes definitely have an effect in combination with the local circulation, and that's what determines the end member composition. The end members are set in their source regions, and it's there the local circulation in combination with the biogeochemistry that sets it. And there's definitely no universal relationship. Changing the end members will also change the correlation. So it's a bit sobering if you're into paleo proxies. But the cadmium and the zinc, you can use them if you have some information to start with. You can, for example, use it to track at a certain time how far North Atlantic deep water and Antarctic intermediate water are flowing north or south. You can't use it to actually calculate nutrient concentrations in the past oceans, as far as I think. And there's also no universal Redfield ratio for metals. There's orders of magnitudes of difference. We know that for iron, we also now see it for cadmium. Depending on how much there is, that will take up more or less. And the influence of local remineralization can significantly influence the relationship, especially if the sampling region is limited. If you had only done the oxygen minimum zone, our interpretation would have been completely different than now that we have this long section where we can see both to the far north and to the far south what's going on. That was it from my side. Happy to answer any questions you might have. And I'll be here tomorrow and the day after, so you can also just be sitting in Tim's office, so you can also just walk off.